Right, we're carrying on with this mineral plant worksheet. So here we have a bacterium. Bacteria is the plural. Okay. Complete the diagram by showing, so they've given the cell wall. So this bit here is the cell wall. And you have to be careful because I missed this bit. It's saying that inside, and it's 3 marks. So if we did the cell membrane, cell surface membrane on the inside. Right, so that's the cell surface membrane or cell membrane. Well, we'll call it cell membrane because you've got your cell wall. So really that's your surface. Right, what do we have? With a bacterium we have this round thing. What's that called? What do we call that? We call that the plasmid. Right, you also get another bit of genetic material. It's like a circular chromosome, or they call it where are we? Nucleoid. Nucleoid. They call it a nucleoid. Yeah, that's it. Right, okay. And then you've got the liquidy part in there. Cytoplasm. So any of these will get you your 3 marks. So if you put 3 of these, you can put more, but it's only a 3 marker. So it's good going through it all, so then it's in your head when you set in the exam. Right, okay. Decomposition by bacteria helps release mineral ions such as nitrates. Right, so decomposers come under what? Composers. There's 2 types that we have to learn. Bacteria and fungi. Explain why the rate of decomposition is affected by the soil of the pH. Explain why the rate of decomposition is affected by pH of the soil, not soil of the pH. Right. What happens with if you've got high pH? Remember, there's this curve. This is pH and this is enzyme activity. Enzyme activity. So at low pH, the enzyme is not active, and then really it should go down here, and I suppose it'll just die. So there's an optimum pH. Just say our bodies, well, it depends which. In the stomach it'll be much lower. So if you've got a pH of 7, you're gonna have maximum activity for this enzyme. That is called its optimum pH. You also have things such as optimum temperature. Optimum temperature. Right, because beyond this, the enzymes become denatured and then they don't function properly. They might function slightly. So we need to write that. So we can say the enzymes will have an optimum pH to work at. If the pH is not at optimum, the enzyme will denature. Basically what happens is you've got your enzyme. I think this is a lock and key model. You've got your product coming in. Under normal conditions, this fits perfectly into there, right? And then it produces a product and that disappears. But at high temperatures or not optimum pHs, what happens is this becomes, or it comes out of shape. So this does not fit in properly. So the rate that you can produce the product, so this is the substrate. And when it's finished, you're going to end up with, just say, 2 products, and they're going to happily go away and do their thing. But when this has been denatured, this is out of shape, and that doesn't fit properly. That's why they denature and reduce the efficiency of the enzyme. Right, now we need to ask, how do nitrates help? Right, I always say nitrate ions will become or digest or turn into amino acids. The plant then turns or uses the amino acids into proteins and DNA for new nuclei or nucleuses. Right, so let's have a look at this. We want to know how nitrate ions get into root cells of plants. So there's your plant, there's your roots, there's the soil, there's your nitrate ions. They need to get into the plant. So if we do a cross section there, there's a cross section of the root, and you've got a root hair cell. It does that. That's your root hair cell. Right. What happens is you've got your nitrate ion, but you've got lots of nitrate ions anywhere in there. So these will not diffuse from high to low because outside, high to low because outside it's low. Inside the concentration is high. Right, and these are too valuable to lose, so you're not going to get flow going out. So what the plant needs to do, it needs somehow to get them in there, and it's by a mechanism called active transport. So we can say the plant uses active transport to take up the nitrate ions from an area of low, so it's opposite to what normally happens in diffusion, to area of low concentration to an area of high concentration of nitrate ions. Now what happens? This needs energy from respiration. Right, let's move on. Right, the graph shows the change in plant growth when different masses of nitrate fertilizer are added to fields. Right. 3.2. On the graph, mark with a cross. Right, limiting factors. Have we done that? Maybe not. So now if you look, if the farmer adds nothing, you're going to get 2,000 kilograms of plant. If he adds 50 kilograms per hectare, it shoots up to 7,200. If he uses just say 75, it goes to 7,600. If he uses 100, it goes to 8,000. If he then carries on adding, it makes no difference whatsoever. So the limiting factor is here. Right, so let's use a different color. Let's use that there. That's where the limiting factor is. So all this here would be wasted. Right. Calculate, oh, I think this is a maths question, but let's have a look. Calculate the percentage increase in plant growth when the mass increases from 50 to 100. So at 50, the plant is 7,200. So look, that there is 7,000. So it's 7,000 kilograms, and then when they go to 100, it goes to 8,000. Oh, 7,000, sorry, 200. And it goes to 8,000 kilograms. So now, can you remember the formula for? It's the difference between the two divided by the original value times 100. So the difference is 8,000 minus 7,200 divided by the original number was 7,200, and then we have to multiply it by 100, and our answer is about 11.1%. So that was a simple maths question. So that's why plants still grow when no nitrate fertilizer is added, right? Because nitrates in different quantities are always in the soil. So nitrates present in soil, right? Also, you have You have nitrogen fixing bacteria that convert. You have nitrogen fixing bacteria in soil. And what does nitrogen fixing bacteria do? It converts nitrogen from the atmosphere, or that's in the soil, back into amino acids. Right, and that's normally done in the roots. So there's a plant. You have these root nodules. Right, and the bacteria live in there. They take nitrogen from the soil and the atmosphere, turn it into amino acids, and in return, what did we say? The plant provides them with food, like carbs. And it's called a symbiotic relationship, because we're the benefiting each other. Right. Sandy soil contains large soil particles with big air spaces, right? So that means it drains easily. Clay soil contains very small particles with small air gaps, are normally filled up with water. Suggests why many plants find it difficult to grow in sandy soil. So first of all, right, the most obvious one is the water will drain away. The water will drain away, and plants need water to help photosynthesize. Secondly, if you've got a solid soil and the roots can establish in there, it keeps it steady. So if there's a storm or somebody brushes past it, it doesn't fall over. So we can say it provides stability to the plant. Yeah. Right, in this, right, I want to undo that. Do you know why? I'm talking about it, but we've got to answer the question. Right. The stability or anchorage in sandy soil is poor. Right, so it's going to become unstable. Use your knowledge of how plant roots absorb mineral ions to suggest why some plants may find it difficult to grow in clay soil. Right, so you've got air spaces in clay soil, but they're filled with water, so there's less oxygen. So less oxygen at the roots means what? Means less aerobic respiration. Right, so if there's less aerobic respiration, that means that this means, and remember, plants absorb minerals using active transport. This means less energy available for active transport. Right, let's keep going. Nitrogen is an essential Nitrogen is essential element for plant growth. Most plants can only use nitrogen in the form of nitrate ions. Only legumes that have bacteria living in their root nodules can use nitrogen from the air. Explain how nitrate ions help plants grow. So nitrates are turned into amino acids which the plant uses for DNA and protein synthesis, or making proteins. Let's use the word making proteins. Right. Name the type of bacteria that live. So they're called nitrifying bacteria. Well, no. Nitrogen fixing bacteria. Because they, what they do is they take nitrogen from the air, turn it into amino acids, and provide it for the plant. Right, many animals excrete urine that contains urea. Some soil microorganisms use the enzymes urease to change urea into ammonium ions and CO2. Describe how ammonium ions can be converted to nitrate ions in the... So remember what we said here. You've got this dead matter. Dead, just say plants. Let's just stick to plants for now. Right. They need to be broken down by decomposers into ammonium ions, yep. And then from there, they get turned into, what did we say? Nitrites, and then into nitrates. Right, so we've got to say right, so what we've got to do is we've got to talk about, let me just, this is what they want you to do. So that's the decomposers. Here and here you've got nitrifying bacteria. Right, so you've got nitrifying bacteria in these 2 points. So they'll turn ammonium into nitrites and then into nitrates. And then the plants can absorb that. So we've got to write that for 2 marks. So we can say nitrifying bacteria. So remember it says, what did it say? Nitrogen fixing, does it say? No. Where was it? Was it up here? No, it doesn't say it. So that there is nitrogen fixing. That's nitrogen fixing bacteria. This here, this bacteria is called nitrifying bacteria, turns ammonium into nitrites and then nitrates, and that'll cover the 2 marks. So that was the wording's got to be spot on for that. So let's just quickly go through that. The dead plant is broken down, decomposed by fungi and bacteria. bacteria. That turns it into ammonium. That ammonium, let's stick to this color. Right, that ammonium is broken down by nitrifying bacteria into nitrites and then nitrates. And then the plant here can absorb that and use it. Okay, that's what they want. Right, 5.2. The graph shows the effect of different temperatures on the rate of urease. You know, the A's normally tells you it's a type of enzyme. What did we say an enzyme was? An enzyme is a biological catalyst. We said a catalyst speeds up reactions. Right, we're not doing the chemistry definition, we're just saying it speeds up the reactions for now. Right, okay. So have you noticed as the temperature goes up, it starts off slow, it goes up, it hits a peak, and then it goes down. This here is the optimum activity there, and this here is the optimum temperature. And then after that, the enzyme starts denaturing and it stops performing. Right, explain the rate of change of urease from 10. So what they want you to do is when you have a let's put it at lower temp. So when you have a lower temperature, at lower temperatures, the molecules or particles have less kinetic energy. Kinetic energy is to do with the movement of a particle, yeah? So it's called kinetic energy. It's to do with the movement of the particle. At low temperatures, the particles have less kinetic energy, so you're going to have less successful collisions. Right. As temperature goes up, as temperature increases, the kinetic energy increases. So you have more successful collisions per unit time. Right. Now explain the rate of change from 36. Right, so we said after the optimum temperature, activity starts going down because the enzymes denature. The active site changes shape. Remember that molecule solo. That fits into there perfectly. A reaction takes place, your product forms, and then it disappears. As the temperature goes up, what happens is this changes. It changes like this. Now that's not going to fit in. It's going to try to fit in, it'll half fit in, so the reactions, the rate of reaction goes down. So that's what we need to write. So we can say at higher temperatures the enzyme is denatured. You've got to use these words, which means the active site changes shape. Right. So the original red thing that's coming in is called the substrate. Once the enzymes acted upon it and the reactions taken place, they are called the things that come out are called the products, yeah? And so the substrate does not fit. Does not fit. There you go. Right. Right now, so we've got this one. We're talking about farmers in Asia add fertilizer to increase the yield. And remember what we said about yield? That's what we said about yield. So for example, the example that we tend to use is this. I plant 10 seeds. 1, 2, 3, 4, 5, 6, 7 grow. So 7 out of 10 have grown. Turn it into percentage, so you multiply it by 100. So I've got a 70% yield. Right, okay, now what we can do is carry on. State the mineral that has the greatest effect on crop yield. No nitrate, no phosphate, no potassium. Nitrate. Right, what they want you to know is when you add a nitrate, the reason it has the greatest effect is because nitrates turn into or converted into amino acids, which are used to synthesize or make proteins, or even DNA on for nuclei or nucleuses. Right. Which crop is most affected by a lack of potassium? Where's potassium? Potassium's there, so it'll be right. So look, if you put no potassium in, what's gonna happen is you're only gonna get a 68% yield. Whilst if you look, lowland rice is not really affected, it doesn't need potassium. And if you look here, if you've got no nitrates, it's going to knock your production right down. Right. So we can call it rye, because if you look in the no potassium, the one with the lowest number is that. Let's keep moving on. suggests why minerals had different effects on lowland rice and upland rice. Right, it's because it's a different species. It's a one marker. Right, you could also have different amounts of or different levels of soil erosion. Right, the yield of mass of the usable part of the crop is measured in kilograms per meter squared. So you've got a big field here, you've got a small field here. What you do is you calculate the mass divided by the total area, and you get the mass, the amount of crop per square meter. You do the same thing here, so then you can compare. So for example, if that is 5.2 kilograms per meter squared of crop, and this is 5.7 kilograms per meter squared, you can see that this smaller field gives a bigger yield per area. Although this might give 5,000 kilograms overall, this might give 205 kilograms, because that's a small area. Per kilogram, you'll per meter squared, you'll get more crop, and it could be for whatever reason. Right, the yield is often measured in dry mass rather than fresh mass. Fresh mass contains water. So for example, look, I've got a field. This year it's been raining heavily. If I take the crop and I weigh it this year, if I look at the mass of it, it could be, just say for example, I don't know, yeah, let's just stick 5.7 kilograms per meter squared. Now next year, if it's drier, right, I've still got the same size of crop, but this time the water vapors evaporated, so I'm going to only get 5.2 kilograms per meter squared. Although the mass is the same. Now if I dried this out here, the water would escape and I would get 5.2, for example. So it's more realistic comparing these two, because this has got no water or very little water. This has got lots of water, so you can't compare the 5.7 to 5.2. That's why what they do is they dry it out. They dry out the crop, so you're not measuring the water. It's like chicken in supermarkets, they add water to it, so you're paying for water really. It's not very ethical, but it happens. Right, we can say here fresh mass includes water. Suggest how the dry mass could be determined. Okay, so what you do is you weigh the sample crop, you place in oven to remove water, weigh again, then put back in oven. Right, weigh again until you reach constant mass, until the mass remains constant. or the same. Right. The table gives data as a percentage. Right, let's finish it off. The table gives data as a percentage of the yield with all minerals present. Explain why percentage is used rather than kilograms per meter squared. Right. In this case, it allows percentage comparison because plants can be different sizes. Plants can be of different sizes or masses. Yeah, so we just want to compare the effect. Where are we? The effect of as a percentage. So what was it originally? By adding the fertilizers, what impact has it had on the percentage? There's a percentage difference. It makes them for a more fair or valid comparison. Okay, let's look at this. Plants need. So remember what we said about magnesium. We said magnesium is used to make chlorophyll, which is in the chloroplast, which is used for photosynthesis to make food for the plant. Okay, so it's the chlorophyll molecule. The chlorophyll, this is the green pigment, the green color. Right. Protein, you need nitrates normally. That's the just best way to do it. Although they'll allow sulfate, ammonium, but nitrate turns into amino acids, which the plant then uses for okay. Right, well, we'll come back. 43 minutes long. Right, it doesn't matter. We'll try to squeeze this in. So let's try to get this question finished in this video. Right, a student uses apparatus to investigate the effect of nitrate ions on the growth of plants. So they're putting in air, which is in effect what? Oxygen for what? Aerobic, what is it called? Respiration. Right. You've got the solution containing the nitrate. And they've got cardboard surrounding. So what they're trying to do is block out the light because of the cardboard. Right. The young plant is grown in a sterile. Why do we have sterile? Right. Number of things. So you don't get any pathogens that cause a disease to the plant, and you don't have any other organisms. You don't have any microorganisms competing. Right. The student repeats the experiment with other young plants, also carries out the same experiment, but without nitrate. Then what they do is they measure the stem of each plant every 5 days. That's fine. So you've got 0, 5, 10, 15, 20, all the way to 55 days. If it's got mineral ions, it grows 23, 30, 45, 98, 145, 160, 163. Without nitrate ions, it grows 23, 25, 30, 38, 38, and then it sort of peaks off and it goes down a bit actually. So that's how important nitrate ions are, because to get growth you need amino acids to make proteins, to make DNA for new nuclei. Right, that's where we are now, yeah? Now look at this question here. Just as to highlight how things are going to change, yeah? So you've got 2 marks, 3 marks, 5, 2 marks, 7, 2 marks, 9. They could have said discuss the experiment, discuss the findings, discuss the students' hypothesis or whatever it was, and then you'd have to figure out, break this down into little bits. But they've done it for you in this question. So they're saying describe the growth of the plants in each solution. So we noticed that with the nitrate ions, the growth was faster. So they both started growing, but the growth was faster. Right. Right, now with the solution without nitrate, the growth leveled out and then decreased. So it stopped growing and then it started decreasing. Let's have a look. So look, it went up, 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 leveled out here, and then started decreasing slightly. That's all we've described. Explain why young plants absorb more mineral ions when air is bubbled through the solutions. Remember what we said about respiration. So oxygen is provided, so aerobic respiration can take place. And what does aerobic respiration do? Aerobic respiration releasing energy. Right, now if you've got mineral ions in the solution, you need to absorb them into the plant, and that's done by a process called active transport. So therefore active transport of mineral ions or minerals can therefore take place. Is that okay? So what you've got oxygen for aerobic respiration, which releases energy, that energy is used by a process called active transport in the roots to take up the mineral ions to help growth. Right. So just why each solution is sterilized. Right. This is to kill two things, right? Remember we said pathogens. What is a pathogen? A pathogen is a microorganism that causes disease. Right, this is to kill pathogens to prevent infection or disease. I mean, to be honest with you, it could be because of to stop other microorganisms. They're not necessarily pathogens to compete for the resources. Right, okay. There was a jar here and it had if you look here, it's got cardboard on. It's to stop light getting in. But why do we need to stop light getting into the bottom part of it? Where are we? There we are. Right. This is to prevent light getting in to stop photosynthesis, to stop photosynthesis of algae, because that'll compete with the plants for resources and might give a poor what do you call it, a poor comparison or unfair comparison. Right, to identify the dependent variable. Right, remember we said there's three types of variable that we look at: independent variable, dependent variable, and a control variable. These are all right. Dependent is the one that you're changing. So in this case, it would be a solution with nitrate or ions and one without ions. Dependent is the one that you're going to measure. In this case, it will be the did you say the number of leaves or length? Yeah, okay, the length. So measure the, in this case, the length of the plant, length. And remember what we said about control variables. This is to help keep the test what? Fair. So for example, your pH levels, amount of water, the temperature, the CO2 levels. These are all things that affect, and light levels. These are all things that affect the growth of a plant. So you need to control those. So the dependent would be the length of plant. Name one biotic. Remember, biotic is living. So bio is living. A bio, as in abiotic, is not living. It's good to know these. Name one biotic variable that should be controlled. One biotic variable that should be controlled. Species. Okay, so these here, all these here are abiotic variables. The other thing that you should control is try to make sure that the age of the plant is the same, the species of the plant is the same. You could make sure that when you start the plant, the size or surface area is the same. Yeah, that'll do it. Right, okay, that's done. That's a long one. That's a super long one, but it'll be worth going through and then trying it on your own because there's a lot of repetition.